Hey guys, hope you are doing great. Today we're gonna discuss about the iron deficiency anemia. Okay, one of the most worldwide nutritional deficiency disease. So during this lecture, first of all, we will talk about the iron metabolism and its absorption. This is the most important part of this topic because if you don't know about the metabolism of iron, you will not approach to the iron deficiency disease or to the patient who has iron deficiency anemia. You will not able to differential diagnose iron deficiency anemia with other types of anemia that they're like iron deficiency anemia. For example, with anemia of inflammatory disease or chronic disease, for example, with thalassemia, for example, with sideroblastic anemia, and so on. And third important importance of this topic or of this iron metabolism is that we have a test by which we diagnose and differential diagnose the iron deficiency anemia, which is called iron profile test. Okay, so to know about the iron profile test, you have to know about the metabolism of iron. Okay, plus we have a protein which is called hepcidin. Okay, we will talk about hepcidin. Hepcidin is one of the most important regulatory protein of iron in our body. Okay, for example, when a patient has chronic inflammatory disease, for example, a patient has inflammatory bowel disease like Crohn disease, like ulcerative colitis. Okay, why they develop iron deficiency anemia. Okay, so that is because of hepcidin. We'll talk about it. So that's why this is the most important part of iron deficiency anemia. So in this part, in this video, we will talk about the iron metabolism, and in next part, in part 2, we will talk about the pathophysiology of iron deficiency anemia. We will talk about the causes of iron deficiency anemia. We will talk about the signs and symptoms of iron deficiency anemia, and also signs and symptoms of iron deficiency anemia is related to the metabolism of iron. That's why iron metabolism is very important. And then we will talk about iron profile test and we will talk about the iron profile test in this lecture. Okay, then we'll talk about diagnosis of iron deficiency anemia. Then we'll talk about differential diagnosis of iron deficiency anemia. And then we'll talk about management and treatment of iron deficiency anemia in deep details, and how to approach to the patient of iron deficiency anemia and how to treat your patient. And then we'll talk about the follow up and some factors. So let's dive into the lecture. Okay, first of all, let me define anemia. Anemia is that when hemoglobin concentration drops below the normal range, okay, or hematocrits drops below the normal range, or RBC counts drops below the normal range. Okay, let me explain that. For example, we'll talk about hemoglobin, that what is hemoglobin or HB, but for now, this is an RBC. Okay, this is, for example, an RBC. This is an RBC. Okay, so inside the RBC, we have a protein which is called hemoglobin, and hemoglobin transports oxygen. Okay, we'll talk about it. Okay, now, when we have less HB in our body or in our RBC, this is defined as anemia. For example, when hemoglobin level drops below the 30 grams per dl in male or below the 12 grams per dl in female. Okay, this is called anemia. Or about hematocrit, listen me carefully. This is blood in a tube. Okay, when we centrifuge it, okay, now blood is made of two parts. Blood is made of two parts. It's made of plasma, it's made of cells. Okay, now about the cells, we have RBC in blood, we have WBC in blood, and we have platelets in blood. But anyway, now this part, this red part of tube, this is made of RBC. Okay, this red part. Okay, and RBC goes down in this tube. Okay, so this part, only RBC part of this tube, of this whole tube, is called hematocrit. Okay, this RBC count, and this are around 45%. Okay, now this green part, this is called buffy coat. Okay, and this is made of WBC and platelets, so this is less than 1%. And this remaining blue part, this one is made of plasma. Okay, that's around 55%. So now this hematocrit level, it means the whole concentration of a RBC, when it drops below the normal, for example, less than 41% in male or less than 36% in female, okay, this is also called anemia. Okay, or when RBC counts drops below the normal, for example, when RBC counts below the drops below 4.5 millions per microliter in male or below 4.1 millions per microliter in female, this is also called anemia. So in a nutshell, simple definition for anemia: when hemoglobin concentration drops below the normal range and hematocrit concentration drops below the normal range, this is defined as anemia. But keep it in your mind that these values are dependent on some factors. Okay, factor number one: high plasma volume or less plasma volume can change this these values. Okay, for example, when you add more volume to the blood here, so you will increase plasma volume, so the percentage of plasma is increased related to the percentage of hematocrit. Okay, so this is not a true anemia. Or in hypovolemic situation, when percentage of plasma is decreased to this area, so now for example your plasma concentration is around 40% and this around 60%, so this is not polycythemia. Okay, so that's why it depends on the volume in our body. Other factors, these values are depending on different ranges in different labs. Okay, in one lab the normal range for hemoglobin is 15 grams per del, and another lab is around 14 grams per del, and so on. It depends on age of the person, it depends on gender, that our patient is male or female, it depends on physiology state. For example, a patient lives in high altitude or in deep areas. Okay, so these were all about the definition of anemia. Now come to the definition of iron deficiency anemia. Now, this is iron. Okay, listen me carefully, this is so easy. This is iron. I'll talk about iron. Iron has many functions in our body. Okay, one of those functions is that it has role in making of hemoglobin. So when we don't have iron, we cannot make hemoglobin. Okay, so when you have less iron, so you will have less hemoglobin. When you have less hemoglobin, so it means you have anemia. Okay, so that when hemoglobin concentration drops below the normal due to iron deficiency, this anemia is called iron deficiency anemia. Okay, so in early stages, iron deficiency anemia causes normocytic, normochromic anemia, but in later stages, it causes microcytic and hypochromic anemia. What do I mean by microcytic and hypochromic anemia? Listen me carefully. This is hemoglobin, okay, inside the RBC. Hemoglobin is an important an essential protein inside the RBC which transfers oxygen to the tissues. Okay. Now, to make hemoglobin, we need iron. This is one point. Okay. When you don't have iron, you cannot make hemoglobin. Now, when you have less hemoglobin, so RBC size decreases. This is one point. In next point, I'll tell you during proliferation that why in iron deficiency anemia the normal size of RBC drops. When the normal size of RBC drops, for example, normal size of RBC is around 90 femtoliters. Okay. Now, when it drops below the 80, below 90, so this is called microcytic. It means the RBC size is small. Okay. And we can read this in MCV. MCV means main corpuscular volume. It means the main volume, the main size of the RBC. So when main corpuscular volume drops below the normal range, it depends on different labs. Okay. For example, below the 80 femtoliters, so we call it microcytic anemia. And why we call it hypochromic anemia? Okay. Hypo because the color of hemoglobin is red, and when you don't produce hemoglobin, so you will have less hemoglobin, and when you have less hemoglobin, so RBC, this normal RBC which is red, it looks pale. Okay. So that's why it don't look, for example, red. So this is called hypochromic. Chromic means color. Okay. So it will lose its natural color which is red. Now let me tell you some things about the iron. Okay. Rather than that iron has role in making of hemoglobin, it has other functions in our body also. Okay. For example, we use iron for DNA replication. These are so important because later on when we talk about signs and symptoms, so we need to know about these things. We need iron for DNA replication. For example, first we need iron in hemoglobin for hemoglobin synthesis. We'll talk about it later on. Then we need iron for DNA replication. For example, this is one cell. I want to make from one cell two cells. So this is DNA inside the cell. So I want to replicate my DNA into two copies. One copy goes to this cell, one copy goes to this cell, and one cell is dividing into two cells. Okay. So now to make from DNA another DNA, this is called DNA replication. So for DNA replication, we need an enzyme which is called ribonucleotide reductase. And this enzyme needs iron. So when we don't have iron, we cannot use this enzyme. We cannot make copies of cell. Okay. We cannot divide our cells. Okay. For example, later on we say that a person who has iron deficiency anemia, it in very advanced stages. Okay. In very advanced stages. Okay. So that person or that patient may develop glossitis. Glossitis means smooth tongue. It means the pili which we have on our tongue, they made up a lot of cells. Okay. So we cannot make a lot of cells, so we will lose these pili from the tongues, and our tongue look smooth. Okay. That's called glossitis. That's why this is so important. And so on, it's also we have a lot of replication of cells in our skin and also in our GI system. The next function of iron is that it has role in ADP production, in energy production. For example, this is mitochondria. This is mitochondria. Okay. So now mitochondria has two cell membranes. Okay. This is outer and this red one is inner. So inside the inner cell membrane of mitochondria, we have a group of proteins which is called electron transport chain. Okay. So now do you know something about ATP synthesis that during ATP production we use NAD, NADH, FADH2, FAD, and so on. So this electron transport chain takes an electron from them and it transports it, and finally it produces energy for us. I'm not going deep inside the physiology, but these electron transport chains. Here we have an enzyme among this, which is called cytochrome B. Okay, so this cytochrome B needs iron. So when you don't have iron, you cannot use cytochrome B. You cannot produce ADP. You cannot produce energy, and you cannot produce a lot of cells. And you will, for example, when a patient of anemia or of iron deficiency anemia comes to you, he says for you, for example, that I'm so tired. Okay, this tired and this weakness is due to less energy production, plus due to less oxygen transportation. We don't have HB. Got it? Then the next function of iron in our body, it has role in many enzymes in our body. Plus, it means in many enzymes in many cellular metabolisms. Okay, this is very important. Neurotransmitter. Do you know something about dopamine? Dopamine in our nervous system, in our central nervous system. Dopamine. This dopamine is made of an amino acid. Okay. Now, when we want to make dopamine, we need enzymes to make dopamine from amino acid. So these enzymes which are going to make dopamine, they need iron. Got it? They need iron. So when we have when we don't have iron, we cannot produce enough dopamine, and it produces a lot of signs and symptoms in iron deficiency anemia. For example, one of those is restless leg syndrome. Okay, patient of iron deficiency anemia cannot rest their legs. Okay, it will change their legs time to time, especially during the night. He senses some pain, he senses some weight in their legs, so he's like this. He's always shaking his foot. Okay, so that's guy restless leg syndrome. He cannot rest his legs. That's why this is so important. And another important, iron has role in collagen synthesis. Okay, do you know signs and symptoms? We'll talk about signs and symptoms of anemia in part two. But one of the iron deficiency anemia sign is an advanced stage of iron deficiency anemia, chylomicria. Okay, the you can watch this photo. This is called chylomicria. So this spoon like nail is due to less collagen synthesis. Okay, and other sign and symptoms, fissures near the oral area also developed due to less collagen synthesis. We'll talk about them in next part. So these were usages of iron in our body. Now, let me tell you something. Our total body iron is around 3 to 4 grams. Around 3 to 4 grams. Okay, our total body iron. Now, from this 3 to 4 gram iron, 65 to 70% of iron is inside the hemoglobin. Okay, it uses for hemoglobin synthesis. It's very important. Around 20 to 25% is in storage form in form of ferritin and hemocyanin. We will talk about them. Around 5 to 10% is inside the myoglobin, a protein inside the skeletal muscle cells, which keeps oxygen to itself, and then uses this oxygen slowly for ADP production. And around 1 to 5% is using for the functions of different enzymes that we talked about them, and around or less than 0.1% is during transportation or during transportation from one part of the body to the other part of the body, and it's in moving uh, fashion. Got it? Now let's talk about absorption of iron. Okay, that how iron is absorbed to our body. Okay, and then we'll talk about hepcidin and so on. Now let's talk about the iron. First of all, first of all, we have in dietary iron. For example, when we take a food, okay, we have, this is called diet. Okay, in diet, when we take a diet, we have two types of iron, which is called dietary iron. One type is called heme iron. Okay, another type is called non-heme iron. Hem means that iron is inside the hemoglobin. Okay, so this type of iron, which is called hem iron, it is Fe2 plus, which is called ferrous form. Ferrous form. Ferrous form. And non-hem iron, it means it's not inside the hemoglobin. This is called ferric form. This is called ferric form. Now, a heme type of iron is available. Heme type, this is for example hemoglobin, this is iron. Heme type iron is available in red meat, okay, in red meat, okay. It's available in fish, for example, and in, for example, in liver and so on. A non-heme type of iron is available in some types of vegetables. Got it? Now, when you take a heme and non-heme iron both into your GI system, or when you eat it, it goes through the esophagus to the stomach. Okay. Now, point number one. Listen me carefully. Point this. I'll talk about these two. Point number one, when you want to absorb iron or extract iron from the food, okay, you need here in the stomach, you need here HCl, you need here HCl, hydrochloric acid. Now, if you don't have HCl in your stomach, you cannot absorb iron properly. For example, when a patient uses PPI drugs for long term, for example, omeprazole, pantoprazole, esomeprazole, lansoprazole. All of them are PPI drugs, and they blocks acid production. Okay, and when a patient is a chlorhydria, it means genetically or due to some diseases, uh, he or she doesn't have enough HCl. Okay, and or in sometimes in chronic gastritis. So in these conditions, we have less HCl and we cannot absorb iron from the foods. Okay, this is one point. Point number one. Point number two. Both heme and non-heme iron come to our GI system. Now, in small intestine, we have three parts: duodenum, jejunum, and ileum. In duodenum, in first part of GI, this iron is absorbed, both heme and non-heme iron. How? First of all, when you take a heme type of iron, only 15 to 35% of this iron will be absorbed. For example, this is an example. Okay, this is an example. When you take 100 milligram irons, okay, only 15 milligrams of that will be absorbed, or only 15 to 30% of that will be absorbed. Okay, it means only 20 milligrams will be absorbed. Or okay, when you take non type of heme iron, okay, only 2 up to 10% will be absorbed. So absorption of non-heme iron is less than the heme iron. Okay, so this is good iron, this is not good iron. Okay, that's why in vegetarians who eat vegetables always, they develop iron deficiency anemia because it's not absorbable very good. Now both of them, heme and non-heme, come here. This is jejunum, this is lumen of jejunum, this is lumen of jejunum. So in jejunum, for example, this is jejunum. These are cells of jejunum. So I'll zoom one cell of jejunum here. So this one, which is near the lumen, this is called apical site, and the other site is called basolateral site. Got it? Basolateral site. Now, inside the lumen, we have this non-heme iron, and also we have a heme type of iron. Okay. Point number one. Point number one. Non-heme. It has an E at the end. Point number one. If you want to absorb non-heme iron, you have to convert non-heme iron first to heme type of iron. So here, if you have an enzyme in apical site of the jejunum, which is called jejunal cytochrome B, or sometimes it's called reductase jejunal cytochrome B. This enzyme converts non-type of heme iron to heme type iron. It means it converts Fe3 plus iron into Fe2 plus iron. Now, this iron, which is called Fe2 plus or heme type of iron, which is converted from Fe3 plus, now this is absorbable. So this will be absorbed into the jejunal cell, and this enzyme is called divalent metal transferase 1. This enzyme is called divalent metal transferase 1. So it will be transported inside the jejunal cell. That's one point. This type of iron is called the heme type of iron. Mechanism for its absorption is not clear, but it's absorbing very easy. But anyway, the protein which transports the heme type of iron from the lumen to the cell, this protein is called HCP. Okay, and this is related to the heme group of iron. Got it? Now both heme and non-heme iron, both of them are absorbing in form of Fe2 plus. In which form? In form of Fe2 plus. Now listen me carefully. Some of this iron, okay, goes to bind to a protein which is called apoprotein, which is called apoferritin, which is called apoferritin. Okay, it binds to apoferritin and it stores inside the different cells of the body in the form of ferritin. So store form of iron is called ferritin. Okay, and sometimes it can be stored in another form, which is non-soluble form, which is very tight packed form of ferritins plus other proteins plus iron, and this is called hemocitrin. This is called hemocitrin. Got it? And when we have more hemocitrin in our body, so this is dangerous for our organs. It causes different organ failures. It causes a disease which is called hemocirrhosis and so on. So this is not good. Okay. Now, some parts of this iron is going to be stored in different types of tissues that I talked, for example, in liver, in macrophages, in bones, in spleen, and so on. Now, some percentage of this absorbed iron is going to enter the blood circulation. Okay, and here we have a beautiful protein. Haha, this is very important. This protein is called ferroportin. This protein is called ferroportin, that which is also inhibited by hepcidin. We'll talk about it. Ferroportin is a protein which transports iron from the apical side to the basal side, and here we have blood circulation. Now iron wants to enter to our blood circulation. Now this is called ferrofourteen. In this also ferrofourteen, so it's transported by ferrofourteen here. Now here we have a cell which is duodenal cell, and here we have blood vessel. Okay, and between blood vessel and our apical cell we have extra cellular area. Okay, this is just intracellular area, this is extracellular area. Keep it in your mind, listen me carefully. Now this iron is transported, this iron is transported from the inside of the cell to the extracellular area by this protein which is called ferroportin. So this is very important protein. Okay, keep it in your mind. Now here, before enter to the blood circulation, this Fe2 plus, which one? Fa2 plus, Fe2 plus. This is converted back to the Fe3 plus, because Fe2 plus cannot be transported by the protein which is called transferrin to the other parts of the body. Okay, we have to convert it again to Fe3 plus, again to ferric form. So this enzyme which converts Fe2 plus to Fe3 plus, this is called hepcidin. This is called hepcidin. Got it? This is called hepcidin. Now we have a soluble protein which is called apotransferrin, which is called apotransferrin. Apotransferrin, I'll write it here. Okay, apotransferrin. This is a protein. Okay, now it acts like a track. It delivers iron from one part of the body to the other parts of the body. Now this iron, which is ferric form Fe3 plus, this will bind to apotransferrin, and in some apotransferrin we have one iron, in some we have two iron molecules. Okay, so now apotransferrin plus iron, both of them are called transferrin. Apotransferrin plus iron is called transferrin. Here apoferritin plus iron is called ferritin. Got it? So now this transferrin, because iron plus apotransferrin is called transferrin, this transferrin protein or molecule with iron, it enters to blood circulation. Now it's ready to go to different tissues, and that we need them. We need some iron for enzymes, we need some iron for making up hemoglobin, we need some iron to go to the central nervous system, we need some irons to go to the bone marrow to make a lot of RBCs. So for example, this is our bone marrow. Okay, and bone marrow, for example, in bone marrow, this is RBC or erythrocyte. So I need iron inside the RBC to make new RBC and to make new hemoglobin. So now how it enters here, because these proteins are so important, because due to mutation of these proteins, a lot of disease will be developed. Okay, so that's why these proteins are so important. So now here, this is this also extracellular area. Now this is transferrin, this is transferrin, this is transferrin. Now this transferrin molecule now wants to enter to the target cell. For example, the target cell here for us will be RBC or erythrocyte, because I'm going to make from one RBC, other RBCs. Okay, so when I want to make from RBC other RBC, I have to duplicate all materials related to this cell. I have to duplicate its hemoglobin also. Okay, now here in target cell, for example in erythrocyte, we have a protein, and this protein is called transferrin receptor 1. This is called transferrin receptor type 1. So transferrin is binding to transferrin receptor type 1. Then it's endocytosis into the cell. Okay, by the help of proteins like clathrin. Okay, it's inhibition internal medicine. So now, when we internalized this transferrin receptor plus transferrin, both of them, into a vesicle, this vesicle is called endosome. This is called endosome. Now, inside the endosome, we have this transferrin plus we have transferrin receptor type 1. Now, by acidic environment where we have a lot of protons, this iron will be disassociated from the transferrin, and now we have free iron. This free iron is then transported into the cytoplasm once again, by a protein which is called uh, dimethyltransferase 1. Okay, now, but anyway, it's transported into the cytosol of the cell. Okay, now listen me carefully. We have which type of iron now? Fe3 plus. We have Fe3 plus. Okay, now absorbable form of iron should be, keep it in your, should be Fe2 plus. Transportable form of iron should be Fe3 plus. And again, usable form of iron should be Fe2 plus. It means I have to convert again Fe3 plus into Fe2 plus. So when I convert Fe3 plus into Fe2 plus, here is an enzyme which is called STIPS 3. Okay, look the abbreviations uh, in your internet. Okay, because you don't need to memorize full length of these names. Okay, but anyway, this is called sex transmembrane epithelial protein, but anyway, you don't need to memorize them. Now, these tips 3 uh, a protein or enzyme, it converts Fe3 plus into Fe2 plus. Now Fe2 plus is available in our cell to use it. Okay, now let me use it in synthesis of hemoglobin. Okay, I'm not going in deep details for synthesis of hemoglobin. Okay, we will talk about all steps of hemoglobin synthesis under the title of hemoglobinopathies or under the title of thalassemia and so on. Okay, but for now, and also I'm not talking in deep details about the steps of erythrocytes production. Okay, and because we don't need it here, we'll talk about it in physiology. But anyway, here the first cell in bone marrow which makes all blood cells, this is called pluripotent hematopoietic stem cell. This is called pluripotent hematopoietic stem cell. Pluri means different, potent means it has capability to differentiate. Pluripotent hematopoietic, hemato means related to blood. Hematopoietic stem cells, it means it can make other cells, and also it can make its own. Okay, its own like new cell. But anyway, pluripotent hematopoietic stems pluripotent hematopoietic stems, it is divided into two cells. Okay, it is divided to myeloid stem cell and lymphoid stem cell. Lymphoid stem cell is going to make B lymphocyte and T lymphocyte. I'm not going in deep details here. We will talk about the WBCs in leukemias and so on. This myeloid stem cell. This myeloid, when we check myeloid stem cell under the microscope, it produces different colonies, more colonies. Colonies means a lot of cells in one area. Okay, one colony is called colony forming unit megakaryocyte, which is going to make platelets. Okay, we'll talk about hematopoietic under the title of hematopoiesis. But anyway, another colony is called colony forming unit monocyte and granulocyte, which is going to make granulocyte like neutrophil, like eosinophil, like basophil, and it's going to make monocyte, which is also called macrophage. And another colony is called colony forming unit erythroblast or erythrocyte. This colony is going to make a lot of RBCs, a lot of red blood cells. Now, from this colony, when we watch it under the microscope, the first cell which will be made from this colony, this cell is called, I'm not going into details, okay, don't worry. This is called erythroblast, okay, because this is very immature, okay. It contains mitochondria, it contains all organelles, it contains nucleus, okay, and it's very large, around 20 micro, 20 micron, I think, okay. This is very large. Now this proerythroblast is then converted to another. I'm going to make one RBC, okay. This is going to convert it to another type of proerythroblast cell, which is called, this blue one, which is called basophilic erythroblast. Basophilic erythroblast. It means we have here now RNAs, a lot of RNAs. Do you know that from DNA, we are going to make RNA, from RNA we are going to make proteins, okay. Now here the most important protein is which one? In RBC, hemoglobin, okay. Now from DNA I'm going to make RNA. So now we have a lot of RNA and some organelles, some ribosomes. So when we stain it, it looks in blue color, and blue color is called basic dyes, or this is called basophilic. It likes basic dyes, okay. Then basophilic cell will be converted to another cell, okay, and this step we convert RNA to proteins. This is the next step. So when we convert RNA to protein, the most important protein in RBC is hemoglobin, and hemoglobin protein has red color. So here in this step, we also use what? Iron. We use iron for hemoglobin synthesis. We use iron. I'll tell you here, but in which a phase. Okay. So now in this step, this is called. This is called. Here we have some proteins plus some RNAs, because all RNAs are not converted to all RNAs are not converted to proteins. Okay, so here if we have two types of dyes, one is due to RNA, which is basic, one is due to hemoglobin, which is red. So this is called polychromatophilic erythroblast. It means here RBC likes a lot of colors. It likes red colors. It likes blue colors. So that's why this is called polychromatophilic erythroblast. Now, here is a point. As much a proerythroblast is dividing, okay, and furthermore it's dividing, the size of RBC is going to be decreased, okay. Size of RBC is decreasing, okay. So now another stage, I'm not going into details, another stage is from which, now another stage after the polychromatophilic erythroblast, here we have only red colors, because all RNA is now converted to proteins, okay, and these protein are called as a collectively hemoglobin, okay. So now hemoglobin has red color, and when we add red color on this RBC, we cannot find red color in red color, okay. So that is why this RBC is called ortho orthochromato ortho just a minute, orthochromatic erythroblast, okay, because it looks red complete. Now in next stage, now this RBC changes its shape now from this round shape to a biconcave shape, and you know about, because this is internal medicine, not physiology, and RBC has a biconcave shape. So now we have some small proteins here, okay, and these small we have some small ribosomes here in some remnants of endoplasmic reticulum and so on. When we add color to it, it looks like a net, okay. Do you know net? Okay, so that's why this is called reticulocytes. Reticulocytes is very important for us, okay. It shows the function of bone marrow. It's very important for us. Now this reticulocyte is ready. Now it is going to the circulation, and after 1 up to 2 days, now it will be converted to a mature RBC, okay, and this whole process takes around 1 week. Now, in these steps, listen me carefully. Why I explained these steps, okay? Because because I said you before that in iron deficiency anemia, we will have less hemoglobin, and we will have less hemoglobin, and due to that, we will have microcytic and hypochromic RBC or anemia. What do I mean by this? Now, listen me carefully. When we have more iron, complete iron source, okay, so RBC will replicate until a mature RBC is produced. But when we have less iron, so less hemoglobin synthesis occur, and when we have less hemoglobin synthesis, less hemoglobin gives signals to this production pathway that I'm not mature, I don't want to go to circulation, lead me to furthermore replicate myself, to furthermore divide myself. So it is dividing further. When we divide it more, its size is decreasing. So that's the cause of microcytic anemia, that in iron deficiency anemia the size of RBC will be small. Plus hypochromic, it means that we will have less hemoglobin per cell. Got it? Now let me tell you something about the hemoglobin, that how we use iron inside the hemoglobin. Okay, not in full details. Listen me carefully. This is mitochondria. Some steps of hemoglobin synthesis is occurred inside the mitochondria, inside the RBC, before RBC is getting matured, and in mature RBC we don't have nucleus, and also we don't have organelles. But anyway, some steps of hemoglobin synthesis is occurred inside the mitochondria. Some are occurred outside the mitochondria in cytosol of RBC. But anyway, glycine, an amino acid, it comes and it joins to succinyl CoA. I'm not going in deep details. Succinyl CoA plus glycine, after many steps, they are going to make a structure, a molecule, which is called protoferrin 9, which is called protoferrin 9. Now, here an enzyme will come which is called ferrochelatase, and this ferrochelatase adds iron Fe2 plus. I said to you before that we need iron here. It adds iron inside or onto protoferrin 9, and as a collectively iron plus which type of iron? Hem type, plus protoporphyrin 9, both of them are called as a collectively hem. Both of them are called hem. Now we will add to him a chain of protein, which is called, this protein is called globin protein, and globin protein plus heme, both of them are called as a collectively hemoglobin. So that's why inside the RBC we have a hemoglobin, and now this hemoglobin is ready to transport oxygen. Okay, inside one RBC we have millions of hemoglobin, and one molecule of one complete hemoglobin is made of 4 molecules of hemoglobins. Okay, and they have different chains. We will talk about them under the title of thalassemia, alpha, beta, and so on. Okay, so now this RBC is ready to transport oxygen. So when we have iron deficiency anemia, we cannot transport oxygen to the body, to the tissues. So we will feel fatigue, we will feel less energy inside our body. Got it? So now, uh, let me explain a bit about the daily absorption and loss of iron. Then we will talk about hepcidin and regulation of iron. Okay. Now, daily absorption of hemoglobin. Okay. For example, when we take a diet, only around 10 to 20 milligrams per day will be absorbed through the gut. Okay. Now, no, sorry, in diet we have only, for example, 10 to 20 milligrams per day in western diets. Okay, only 1 to 2 milligrams will be absorbed because we lose only 1 up to 2 milligrams of iron through different processes in our body, through feces, for example, through epithelial cells, through sweating, through for example, menstrual cycle in female. Okay, through different pathways. Okay, it means we don't have active system to excrete extra iron from the body. That's why iron absorption and excretion is completely regulated. Okay, absolutely regulated, because when we absorb more iron, we don't have a process to excrete it actively, so it's converted, more iron is converted to hemocydin. And when hemocydin is accumulated in our body, it's called hemosiderosis. And hemosiderosis causes a lot of disease. It causes heart failure, it causes kidney failure, it causes a lot of disease. That's why iron absorption is tightly regulated, and it's the under the control of hepcidin. We'll talk about it. Okay, this is the most important part, because daily we use around 20 to 25 milligrams, okay, per day. We use around 20 to 25 milligrams iron per day, but we absorb only 1 up to 2 milligrams per day. Okay, here is the point: 20 up to 25 milligram of iron is recycled by macrophages every day. Is recycled. This is the most important part of iron that is provided to our tissues daily. Okay, because daily we use 20 to 25 milligram iron, but we absorb only 1 up to 2 milligram. So this other 23, 22 milligrams come from recycling of iron by the macrophages. Let me explain this here. Come here. After 120 days, when RBC is getting older, okay, RBC has a biconcave shape. With it's getting older, it loses its structure of its membrane, and it's converted to a spherical, to a round RBC. And this is now old RBC. And we have spleen. Inside the spleen we have very thin capillaries, which are called cords of Bellrath. But anyway, inside the spleen, for example, their capillaries. Okay, I'm not going to talk much about this, because we'll talk about this under the title of hemolytic anemia. But anyway, inside the spleen, here these are thin capillaries. Okay, now this is macrophage inside the spleen. Okay, this macrophage is here ready to eat RBC. Which RBC? RBCs which are old, which are round. They will stick there. When they come here, they will stick there. And a normal RBC, which has a biconcave shape, it has a flexible shape. It will pass through these areas. But this round RBC, this old RBC, after 120 days, it gets stuck here. Okay, now macrophage is eating that old RBC. Now inside the macrophage, this is inside the macrophage. Inside the macrophage, this RBC is dividing into two parts. Okay, it's dividing into cell membrane, and it's dividing into hemoglobin. And cell membrane is made of amino acid. It's made of fatty acids, and so on. It will be divided into amino acid, fatty acids, and so on. The hemoglobin part, hemoglobin, this is hemoglobin part, HB part. It is dividing into two parts. Okay, one is called a hem part, a hem part. One is called globin part because it was made of globin and hem. This globin part is dividing again into the amino acids. Okay, now hem hem part is separating into two parts. Okay, it's separating to a pyrrole things or protoporphyrin, and it after many steps it's converted to bilirubin, and that's why in hemolytic anemia, due to hemolysis of a lot of RBC, more bilirubin is produced, and more bilirubin causes jaundice because bilirubin has yellow color and it deposits in sclera of our eye, in skin, and so on. We'll talk about it under the title of hemolytic anemia. Now we remain about, because heme is divided into uh, protoporphyrin and so on, or into pyrrole things, and then it's converted to bilirubin. And the next part of him is this iron, this iron. Okay, now this iron enters to the blood circulation. Okay, and it's transported by transferrin again, and it's recycled and it's ready to be used, and that's about 20 to 25 milligrams per day. Got it? And also this iron is Fe2 plus. When we transport it by transferrin, we need it to again convert it to Fe3 plus, and it's also converted by a protein which is called ceruloplasmine. Ceruloplasmine, and the protein ceruloplasmine, and another protein which we discussed here previously. What was this? This protein which is called hepcidin. Both of them are doing one job. Got it? So this was all about absorption of the iron. Now we will talk about hepcidin, why inflammation affects iron metabolism, and also we will talk about iron profile test. Great. Now let me talk about an important protein which is called hepcidin. Okay, this is very important in controlling and absorption of iron. Okay, let me explain it. This is a cell. Okay, this cell is related to liver. Okay, because the protein which is called hepcidin, it's released by the cells of the liver. Okay, so it's a liver protein. So let me explain its mechanism because it's very important. Okay, now listen me carefully, this is too easy. When we have more iron, for example, when we have, this is a di, okay, when we have, sometimes we have two irons in transferring, sometimes we have only one iron. So when we have more iron inside our body, okay, or inside the circulation, okay, it stimulates this high amount of iron. Listen me carefully. It stimulates some cells inside our liver which is called liver sinusoidal endothelial cells. Okay, don't memorize long term, only memorize shortcut form. Liver sinusoidal endothelial cells will be stimulated by high amount of iron. Okay, now this one point. So these LSECS cells, okay, they excrete some molecules, okay, and these molecules are called BMP, or sometimes it's called bone marrow morphogenic proteins. But anyway, this is called BMP. Okay, now plus 1 point, high iron causes production of more BMP in our liver. Okay, plus high concentration of iron stimulates a receptor on our hepatocyte which is called transferrin type 1, and it is then transported here into the cytoplasm by transferrin type 1. Oh yeah, we talked about it here. By transferrin type 1 into the cytosol. Now this high concentration of iron stimulates the DNA and genes in hepatocytes to produce some receptors and express some receptors on the cell membrane, which are called BMP receptors. BMP receptors for this BMP molecule. So now, in iron during iron load situation or during high concentration of iron, we have a lot of BMP which are secreted by liver cells, and we have a lot of BMP receptors for these BMP which are also expressed on the cells of hepatocytes. Now these BMP molecules come here and these stimulate their receptors. Okay, now this is one point. Point number two, when we stimulate transferrin receptor 1, so iron is going from the outside of the cell to the inside of the cell. Plus here near the transferrin type 1 receptor, we have another protein which is called HFP. Or so now this HFE is disassociated from transferrin type 1 receptor when it's binding to the iron. Okay, so now this HFE protein, okay, it also stimulates another type of transferrin receptor which is called transferrin type 2 receptor. So now transferrin type 2 receptor and BMP receptors, both of them generates a signal under specifically BMP receptor type 2 and type 1. It phosphorylates here inside the cytoplasm a beautiful protein which is called SMAD protein type 1, 5, and 8. SMAD protein. Okay, now or suppressor of mother antigenic decapenta uh, globin like this. Okay, so this is a SMAD protein 158 type plus SMAD4 protein. Both of them are joining together. So now SMAD158 is phosphorylated. It's phosphorylated by BMPR receptor signals, and also it's now joining to SMAD4. So complex of SMAD158 and SMAD4, both of them go and translocate inside the nucleus. Okay, and these stimulate a specific region, a promoter region, an enhancer region inside the DNA. Okay, which promotes HMP, which the gene which is responsible for production of hepcidin, HMP promoter area. Okay, so now HMP promoter area produces a lot of RNA, and this RNA is then translated to a lot of protein, and this protein would be hepcidin. Okay, so now this hepcidin, this high amount of hepcidin, this is the most important part. Okay, listen me carefully. This high amount of hepcidin comes here to the enterocytes, and do you know here inside the enterocyte, it degrades the protein which is called ferroprotein, and it suppress the expression of this protein which is called ferroprotein, and now we don't have, or we have less amount of ferroprotein, and we cannot transport iron to the blood, we cannot absorb iron into the blood. Now do you know, during inflammation, come here, during inflammation, when a person has long-term inflammation, a lot of interleukins proteins are produced, and one of those is interleukin 6. Interleukin 6 also has receptor here on this hepatocyte which is called interleukin 6 receptor. So interleukin 6 stimulates interleukin 6 receptor, and it also generates a pathway which is called state 3 pathway. And due to this pathway, this gene also be stimulated, which is called state or E region, okay, or responsive area. And if this is BMP responsive area, okay. So now state 3 plus high iron, both of them stimulates promoter regions of HAMP region, okay, and they will produce a lot of hepcidin. So that's why when you have long-term inflammation, so your liver cells produce a lot of hepcidin. So this high hepcidin levels prevents from absorption of iron. So that's why in long-term inflammation you will develop iron deficiency anemia, okay, got it. And here, when you have less iron, okay, I'm talking reverse. When you have less iron, so less transferrin receptor will be saturated. Protein will not be disassociated from this point. It will not stimulate transferrin 2 receptor. And also when you have less iron, the liver cells cannot produce BMP, okay. And when you don't have BMP, plus when you don't have iron, so you will not have BMP receptor. You cannot stimulate this SMAT pathway. You cannot stimulate this BMP RE region, and you cannot stimulate HAMP promoter area, and you will not produce a hepcidin. And when you have less level of hepcidin, so hepcidin cannot degrade ferroportin, so you will have high ferroportin level and you will absorb much iron. This is the metabolism of hepcidin, okay, got it. Let me talk in a brief details. We will talk in full details in part 2, in brief details about iron profile test. For example, a patient comes to you, and in next lecture we will talk that how you diagnose, how you approach, how you treat iron deficiency anemia patient. But anyway, you gave him a test which is called iron profile test. In this test, you will read first serum iron, okay. This serum iron, when we read serum iron, this iron is not in free form, because free iron is toxic. It makes ROS, activates ROS system, and ROS is a toxic system for our cells, for our body. But anyway, now this serum iron is, which we measure in iron profile test, it is saturated by transferrin receptors. This iron is in transferrin form, not in free form, got it. And this is around 60 to 170 microgram per dl, okay. And when we have less serum iron, it means we have iron deficiency anemia. But listen me carefully, certain situations uh, uh, has direct and indirect effects on these profile tests. But anyway, the next one is total iron binding capacity. Listen me carefully, this is very important. When you have high concentration iron inside the circulation, so all these transferrins will be saturated by iron, okay. Now we have less iron, less transferrin or apotransferrin to transport iron, because all of them are saturated by iron. It means we have a lot of trucks, okay, that they deliver cargo and things to an area, okay. When you load all trucks, so you will not have empty truck to transport furthermore things, okay. So that's why this is called total iron binding capacity. It means when you have high concentration iron, all transferrin will be saturated, okay, and total iron binding capacity will be less. It means I don't have much space to transport iron. That's why in iron deficiency anemia, total iron binding capacity increases, okay, because we have a lot of empty tracks. But in normal situation, we have around 250 up to 450 micrograms per dl, okay. Now let me talk about transferrin saturation. Transferrin saturation explains us, okay, that how much percentage of this transferrin is saturated by iron. Like it's reverse of total iron binding capacity, okay. So for example, how we can find transferrin saturation? We will take serum iron and we will divide it by total iron binding capacity, and then we will multiply it into 100, okay. The range below, the normal range would be 20 to 45%. The range below of 15 or 16% is defined as iron deficiency anemia. Got it? It means that these receptors are not fully saturated and we don't have much iron, okay. Number four are about transferrin receptors, okay. This is what is here, okay. Okay, about soluble, yes, this soluble transferrin receptor, this transferrin receptors which enters iron into the cytoplasm. Do you know when an RBC is developing, it expresses millions of these transferrin receptors on its cell membrane, millions, okay. So now when we have less iron in body, or a patient who has iron deficiency anemia, so this erythrocytes or erythroblasts try to take a lot of iron. It's angry, okay, so it produces and expresses a lot of transferrin receptors, and some of these transferrin receptors are shedding into the circulation. They come out into the circulation. So normal amount of transferrin receptor which is soluble, which is in our circulatory system, is around 0.8 to 2 milligram per liter, okay. About so in iron deficiency anemia, this soluble transferrin receptors are increasing. Got it? About CRP, this is also important. When you give iron profile test, try to give CRP test also, because C-reactive proteins, they increase during inflammation, okay. So its normal value is less than 5 milligram per liter, okay. And a lot of time we will talk about it in part 2. A lot of time beside the iron deficiency anemia, we check for the causes of iron deficiency anemia, because anemia is like a sign and symptom, so there must be a cause behind it. You have to treat both of them. You have to treat anemia, plus you have to treat the cause of anemia. So when you are searching for the cause of anemia, so you can find these by one of those way will be CRP or C reactive proteins, okay. These are all about the iron metabolism, okay. This is very important. If you don't know about the iron metabolism, you will not know about how to approach to the patient of iron deficiency anemia, how to treat it, how to manage it, how to know the cause of the iron deficiency anemia. So have a good time till next lecture. Please subscribe and like our YouTube channel. Have a good time.